Hi, my name is Berger, and today I'm going to talk about the memory, boosting and duration of immunity. As an overview on today's talk, I want to first introduce what is memory and then talk about what happens long-term with the immune response, and then focus practically on examples how we can enhance and prolong immunity, such as booster doses, mechanisms of conjugated vaccines, vaccine adjuvants, or vector vaccines that can activate immunity more broadly. So why is memory important? The basis of immunological memory is that upon a second encounter with the same antigen, we can respond more rapidly and more rigorously to the threat. This is also the concept of vaccination, as depicted in this example. If we look at the antigen A, which could be a vaccine, for example, hepatitis B surface antigen, and we vaccinate somebody with this after a few days to weeks, we find a primary anti-A antibody response. Since we only vaccinated with anti-A, we only find a response to anti-A. If in the future we encounter the same antigen, but here in the example of hepatitis, for example, natural infection, where there's not only the surface but also the core antigen present, we mount an immune response against the previously vaccinated antigen of a much stronger antibody titer that also rises much quicker. This is the function of memory, that we are prepared and can respond quicker. So let's look at this in more detail. Here, depicted on the left side, we have 3 different B cells. One that recognizes a red antigen in the middle, and 2 that recognize different antigens. If you have a natural infection or a vaccination with an antigen, here in red, it binds to the red B cell. This B cell starts to proliferate and to form clones. So part of these clones then get transformed into plasma cells that are able to produce a massive amount of antibodies that fight the infection. However, there's also another part of the B cells that goes into the line of memory B cell formation. So these cells, they don't produce that many antibodies, but they can survive for a long period of time. And while the plasma cells go to the bone marrow and reside there, the B cells patrol the body, and in case of a reinfection with the same pathogen, they are able to very quickly respond, to proliferate again, and to form many more plasma cells out of one B memory cell. This explains why the secondary response is more rapid and stronger. So now we can wonder how long this memory that we induced by a vaccination or natural infection can last. And in general, natural infection induces lifelong immunity. The exception is maybe that at older age we undergo immunosenescence and thereby might become susceptible again. But if we look at only the time period from, let's say, up to 60 years old, in general, natural infection induces lifelong immunity. The same is true for live vaccines, as depicted here on the right side in the example of measles. So that you understand this graph, I have to give a short introduction. So in this very interesting study from Amana, published in the New England Journal of Medicine, they analyzed lab workers. There was a primate research facility that have been given blood over many years. And they tested how their anti-vaccine titer and also the titers against natural infections evolved over time. So each colored line here represents one of these lab workers, and it's plotted on the x-axis on the age of the worker at the first measurement as well as on the last measurement. And analyzing many of these workers, they found that, for example, the immunity against measles vaccination, which is a live vaccine, lasted virtually forever. So mathematically, they could calculate the half-life time of these antibodies, which was about 3,000 years. This is in contrast to tetanus vaccinated subjects, where they calculated only a half-life of about 11 years. So you see that the arrow declines, so over time the immune response against tetanus vanishes. This is of course well known, and this study just nicely put it in context of natural infection and then tested it for different antigens that I also haven't shown here. So if we know that live vaccines give lifelong immunity, but for example protein vaccines such as tetanus vaccination don't give lifelong immunity, it's obvious that we try to find ways how we can boost this memory. And this is just an overview on what methods or what mechanisms have been tested or are in clinics. So memory can be improved by different ways that I want to quickly touch on here. So one is booster doses, another one is conjugated vaccine, then the use of adjuvants or the use of vector vaccines. Some of these topics are also covered in other lessons of this course. The probably most important and most known way how you can increase memory generation is the application of booster strategies. Boosting means that the same antigen is repeatedly given to the same individual, and with each administered dose, the antibody response or the immune response in general augments. This is an analogy to the slide that I showed in the beginning of this presentation. So if you look at this example of patients, or in this case it was not patients, it was persons who had to go to prison where there is a high risk for hepatitis B infection, they needed to be vaccinated. And the usually administered interval for hepatitis B primary immunization is a scheme of 1 month after the first dose, the second is given, and then 6 months after the first dose, the third immunization is given. And this is here represented in the thick black line, where we see that after about 1.5 to 2 months, the line crosses the HBs antibody titer of 10, which means that after that time point, the people could be considered protected to some level. And then after the third administration of the vaccine, the peak titer went even higher. In contrast, if you shorten the interval of the immunization, which is here depicted with the blue arrows or the dotted line, and this is a so-called rapid immunization scheme, then you more rapidly induce levels that are above the threshold for protection. So already at 1 month, most of the vaccinated subjects were above 10 units per liter. But you see that at the end, the peak response is lower. And that's why persons who get a rapid vaccination scheme need a last boost after 1 year generally. So this example illustrates that with booster doses we can increase the overall peak response, and at the same time it shows nicely that it's important how we choose the intervals between different vaccinations. And one of the explanations is that after antigen exposure, the cells, they start to clonally expand, as we have seen before, and produce antibodies. And after initial phase of expansion, these cells contract again, so they reduce their number. And if we vaccinate in that specific time where the cells are contracting, then the immune response is less strong than as if we wait for a longer period of time, which explains why for most vaccinations we wait 4 weeks between doses. Another study that I want to show here highlights that the dose we give in a single boost can also affect the effectiveness of the vaccination. So this is a study that has been performed in HIV-infected individuals. All of them were immunocompetent, so they had CD4 T-cell counts above 200. And they were either vaccinated with the standard dose of 3 times 20 microgram intramuscularly given, so we talk about hepatitis B vaccination again, or they received 4 times the double dose intramuscularly, so 40 microgram of the antigen. And there was a third arm that received intradermal vaccination. And if we look then at the immune responses achieved over time, we see that the geometric mean titers, they increase in all 3 groups, but the intramuscular 20 microgram group, which is the lowest one with the blue arrow indicated here, have the lowest peaks. Whereas the intramuscular with the double dose not only reached the highest peaks, but also had the most rapid increase in mean titers. In the setting of HIV infection, this also resulted in that more vaccinated subjects achieved overall protection through the vaccine as compared to standard scheme. But I just want to highlight that the dose, in addition to the interval, is important in achieving a strong immune response. So booster doses can be given at different intervals or at different doses. So a second problem in general in vaccination, and specifically in patients that are immunocompromised or in young children where the immune system is not mature enough, is that we need T-cell mediated help in order to generate strong B-cell vaccine responses or autoimmune or immune responses. So this is a scheme from the previous lesson where we see that the vaccine antigen can activate B cells, give the signal 1, but also activate T cells, CD4 T helper cells that provide a second signal that then results in the maturation of B cells to plasma cells and memory B cells. If we have a vaccine that is not a protein antigen, then it cannot be processed by dendritic cells and cannot be presented to naive T cells. And the classical setting where we have this is if we vaccinate with polysaccharide vaccine, as it's the case for meningococcal vaccine or pneumococcal vaccine as the most prominent examples. And here, when we see that the polysaccharide chain can activate B cell receptors on the surface of B cells, this can lead into differentiation of plasma cells, but there is no strong memory formation. And therefore, the immune response is less strong and also lasts less long. The antibodies that are produced are mostly IgG2 and IgM. So how to overcome this problem? One way is the use of so-called conjugated vaccine. And a conjugated vaccine is the same polysaccharide chain, but this time linked to a carrier protein, here depicted in blue. And this carrier protein can be taken up by the B cell and be presented to T cells, which can provide help to the B cells by secreting cytokines. So this additional signal allows the generation of memory responses of memory B cells, depicted on the bottom in red, and at the same time also the polysaccharide-specific plasma cells, which are able to do a class switch due to the help by T cells and produce more IgG1, IgG3, and also IgA. So this is exemplified in the use of the pneumococcal vaccine that is either available as a polysaccharide vaccine or a conjugated polysaccharide vaccine. And here we just compare these two. On the left side, the polysaccharide, on the right side, the conjugated vaccine. And this summarizes what I mentioned before. So the conjugated vaccine has in addition a diphtheria toxoid protein that allows the activation of T cells. And this additional T cell dependent enhancement of the immune response increases the class switch, generates memory formation, and also allows the establishment of a mucosal immunity, which is crucial in order to reduce carrier of specific bacteria or viruses, and also generate a strong response at the borders to the outer world so that the bacteria or viruses cannot evade the body. And in the setting of pediatric research, this is specifically important because only with this additional T cell help we were able to induce good immune responses in children. As with the polysaccharide alone, the vaccine is ineffective in children below 2 years old, and also many non-responders occurred. A second way how we can enhance the memory formation and also broaden the immune response is the use of adjuvants. So the immunological adjuvants, they act through different ways. And here exemplified is in blue depicted the AS04, the MF59 or alum, which are all currently used in some vaccine preparations. They act in different ways. For example, AS04 is believed to stimulate toll-like receptors on the surface of dendritic cells, and this induces the activation of the dendritic cell that produces more cytokines that enhances the T helper response and thereby enhances the immune response of B cells, better memory, longer lasting memory. Alum has a similar effect, although it's not that well understood. So it can act through activation of the inflammasome, used caspase activation and the secretion of pro-inflammatory cytokines. The MF59 is believed that it mostly works on the enhanced uptake of the antigen into the antigen-presenting cell and thereby modifying the antigen processing steps. This ultimately results in the better presentation and better priming of naive CD4 T cells, and in turn, as we learned before, this is crucial in order to help B cells to establish a long-lasting and strong immune response. So the adjuvants are also one way how we try to artificially mimic natural infection, because in natural infection, this is what happens. Toll-like receptors are activated, antigen presentation is facilitated by general activation of dendritic cells. There's different proteins that are expressed as well as polysaccharides, and this is just an attempt to achieve this using the adjuvants. A more modern approach takes advantage of so-called vector vaccines, and thereby we can introduce the DNA of a protein that encodes a protein that we want to vaccinate against into a live vector, which is usually a virus that doesn't cause disease in humans or that we had immunity already before. So examples are the human parainfluenza virus or replication deficient adenovirus vectors or vesicular stomatitis virus. These viruses are genetically modified to express the gene of interest and then result in the production of the protein we want to vaccinate against. And if we inject that into a vaccinee, they start to replicate, activate the different layers of the immune system, including innate immunity, and can thereby lead to a much longer lasting memory similar to those seen in live vaccines. This is something that is probably more in the future, but has gained some media attention because of the current Ebola outbreak, or the recent Ebola outbreak, where the two vaccines that have been tested both consisted of either adenovirus or vesicular stomatitis virus vectors. Another advantage of a live or a vector vaccine is that we don't only induce B cell responses and CD4 T help responses, but we can also induce CD8 T cell responses. And the CD8 T cells are the cytotoxic T cells able to kill virally infected cells. The response to these can be measured with peptide MHC tetramer. This is just an example here. And they are classically found in live vaccines. So this is an example showing the immune response against MHC-A2 restricted peptide of yellow fever following yellow fever vaccination. Up to 6% of the T cells respond to this peptide. So live vaccines induce strong CD8 T cell responses, as do vector vaccines or vaccines with strong immunoadjuvants. As take-home message, I hope I could show you that live vaccines induce very robust memory similar to natural infection, and for this, broad activation of the different arms of the immune system are important. Together, this ensures that also T-cell memory is established and that mucosal protection exists. Booster doses, thereby the interval or the dose itself is important, as well as protein conjugation, the use of adjuvants, or the use of vaccine vectors can be applied to enhance the immune response by mimicking natural infection or re-exposure.